Hello and welcome, everyone. This is SFU's Café Scientifique. Thank you so much for joining us tonight. My name is Cynthia Henson, and I will be your host for tonight. At this time, we will give it a minute or so for more people to come into the room. But thank you so much for joining us at this time. So many familiar names. Thank you so much for always joining us throughout this series. And a warm welcome and hello to all those joining us for the first time. We do have had some registrants from out of SFU and out of BC, in fact, and thank you so much for joining us. This is Simon Fraser University's Café Scientifique. And it's actually our last session for the spring term, our fourth session for the year. So thank you for being with us tonight. All right, I have 5:02 on the time. I guess we better get started. Again, hello and welcome, everyone. My name is Cynthia Henson, and I will be your host for tonight. This is Simon Fraser University's Café Scientifique. This is a discussion series where we do feature our faculty presenters from the Faculty of Science. And this is our fourth session for the year and the last one for the spring term. After tonight, we will have our next session in the fall term, and we stay silent for the summer. But we try to hold at the very least 6 sessions a year, and we're staying virtual for now. Just know that after tonight's event, we will be sending out a feedback link through the emails that you have provided us. And we do appreciate if you could fill out that web survey and let us know any comments or suggestions that you might have about this discussion series. Your emails have also been included in our mailing list, so you should be receiving updates on the next faculty events even throughout the fall term. We're happy to see all the Café Scientifique regulars. Thank you so much. I see lots of familiar names, and a warm hello to all those joining us for the first time. I hope you keep joining us in the next future events as well. And it's also great to see lots of our community partners join us each time we do this. Thank you so much. Let us begin by acknowledging that we're privileged to gather today on the unceded traditional territories of the Squamish, Tsleil-Waututh, Coquitlam, and Musqueam peoples. We thank them for having cared for these lands and waters since time out of mind, and look forward to working with them in partnership as we continue to build this great city together. If you know whose territory you're currently on, please take a moment to reflect on this. And if you don't know whose land you're on, I encourage you to look it up and make yourself familiar with the indigenous peoples whose land you occupy. All right, just a few house rules before I turn it over to our presenter for tonight. There is a Q&A box on your screen. You have access to that. We have turned off the chat page for now, just to keep things simple. But do use the Q&A box to type in any questions or comments you have about our presentation. There's also a live transcript that is turned on for your convenience if you find that helpful. And also just a reminder, this event is being recorded, and we will upload the recording on our Faculty of Science YouTube channel after tonight's event. All right, as you see on your screen tonight, we have a faculty presenter from our Department of Statistics and Actuarial Science, Dr. Lloyd Elliott. Title of his presentation is The Pathways from Our DNA to Our Brain. From early development all the way to aging, DNA affects the structure and function of our brain. Mapping these pathways helps scientists understand how our brain works and how to fight neurodegenerative diseases. Dr. Lloyd Elliott is an assistant professor in the Department of Statistics and Actuarial Science, and he has provided statistical support to infectious disease researchers across Canada. We're so happy that he agreed to present for tonight. So at this time, I would invite Lloyd to turn on or to screen share your slides, and I'll hand it over to you. There you go. Thank you.
Thank you, Cynthia, for that kind introduction, and welcome everyone to Café Scientifique. So my talk is on the pathways from our DNA to our brain. And I'm going to start by talking a bit about myself and my research. I'm in the Department of Statistics and Actuarial Science here at SFU, and I'm a Michael Smith Health Research BC Scholar Award winner. And my research is funded by Michael Smith Health Research BC as well as NSERC. And my work covers three main areas. In the first area, I'm looking at brain imaging genetics. So looking at how human genetic variation affects the structure and function of the brain. After the COVID-19 pandemic began, I started to do work in epidemiology and host genetics. And in another line of work, I do theoretical machine learning, including developing algorithms and methods for deep learning and other aspects of data science. I named my lab the Statistical Support Team after the COVID-19 pandemic came, as I started to shift focus to support the efforts of epidemiologists across BC, in particular here at SFU in the math department. We have the MAGPIE group. So MAGPIE stands for Mathematics, Genomics, and Prediction in Infection and Evolution. So this group has done a lot of work for modeling COVID-19, including projection of case counts, as well as estimating the number of unrecorded cases. And as a statistician, when the pandemic came, I began to offer my support to MAGPIE and other groups of researchers. For example, the BC COVID-19 group, which is a group of academics at University of Victoria and UBC, as well as SFU, and they're throughout the pandemic they were in contact with BC CDC and with other decision makers. So that's where the name of my lab is from, through supporting the efforts that we were doing at SFU. And in my talk today, I'm going to start with an introduction to genetics. So this is going to be really from the beginning. So for many of you, it's things you've already learned before, but I'm going to maybe give a bit more of a modern spin on it, as well as highlight the things that are important for understanding how genetics are used to look at brain imaging or to look at COVID-19. And to illustrate the ideas I'm going to go over in this introduction, I'm going to focus on an example with breast cancer. And then after that, I'm going to show how this breast cancer example can be sort of generalized to many genes and many different phenotypes and diseases. And then I'll talk about COVID-19 host genetics and brain imaging genetics. So these are two application areas that I've been involved in through my work at SFU. So what is DNA? DNA is genetic material. It's what's passed down from parents to children. So for humans, our DNA is 3.1 billion base pairs long and is organized into 23 pairs of chromosomes. So we have chromosomes 1 until 22, and then we have sex chromosomes. And the DNA itself is a long molecule made up of these base pairs, and its shape is a double helix. So there's two strands of DNA that wrap around each other, like these snakes in this ancient symbol of medicine. So interestingly, this symbol is often used for medicine, but this is from Greek mythology. So this was this was used before we knew the double helix nature of DNA. But on the right, you see a photograph of the discovery of the double helix. So this is a photograph by Rosalind Franklin, a UK researcher who. So she took the first pictures of X-ray diffraction patterns of DNA, and from that proved that DNA was a double helix. So this is the research that led to the Nobel Prize in the discovery of the shape of DNA. But what's important for us here? What's important for statistics? It's the linear and discrete aspect of DNA, which means that DNA can be written as a string, so as a sequence of characters. In this example here, so on the last line of this slide, I have the first characters from chromosome 1, according to a reference sequence of the human genome from the National Center for Bioinformatics. So what this is saying is most humans at the beginning of chromosome 1, they'll have the base pair T, A, C, C, T, A, C. And then for chromosome 1, we have an additional hundreds of millions of base pairs after these. So each base pair has one of 4 possibilities, which are coded as ATGC. So that's for adenine, cytosine, guanine, thymine. These are the sort of rungs of the double helix. So if you imagine these two snakes, there's sort of ladder rungs between them. Each rung is a different letter, and there's four possibilities for the letter. So in the end, chromosome is a very long word, and the word, each letter of the word has 4 possibilities. So that's what's important for statistics, the fact that in the end there's a discrete structure. So we have A, and then we have another A in between that. With DNA, there's no additional information that's hidden between these two adjacent base pairs. Most of the time, you could consider there might be some exception if we consider epigenetics, but at the basic level, all we see for DNA is this long string of letters. And what do those letters mean? So the reason these base pairs are important, and the reason DNA is important, is because of genes, the genes in the DNA. Everything that makes up our body is either a protein. So by mass, we're 15% protein, but on average. Or so everything in our body either is a protein, or it's actually made by proteins, or in some way operated on by proteins. So if we could consider what's in our body other than protein, so there's fat, water, oxygen, CO2. So when oxygen moves around our body, it's proteins that are doing that. When water moves in and out of the cell, it's proteins that are modulating that. So proteins are doing essentially everything in our body, and DNA has the blueprint for all our proteins. So the sequence of base pairs that we see in the DNA, that's translated in a few steps, finally into a protein. So the DNA, it has the blueprints for the protein, and also it controls how much of each protein is made. And this, how much of each protein is made is going to potentially change based on the tissue or based on the context, like what the body is doing. If we're exercising or thinking, then the expression levels of proteins in our cells are going to be different. But essentially everything that we're doing and it is coming originally from the DNA of our cells. So here's an example with the BRCA1 and BRCA2 genes. So these genes are on chromosome 17. So BRCA1 and BRCA2, these acronyms stand for breast cancer type 1 and type 2 susceptibility protein. And these genes are associated with breast cancer. But everyone has these genes. So usually on chromosome 17, someone would have the BRCA1 and BRCA2 genes. It's not the genes themselves that are causing cancer. In fact, it's sort of the opposite. The job of the gene, the job of the protein coded by the BRCA1 and BRCA2 gene is to repair DNA and to destroy cells with DNA errors. So BRCA1 and BRCA2 are actually trying to prevent cancer. That's the job of those genes. That's the job of those proteins. Also, on the lower right here, I have a 3D rendering of the BRCA1 protein from AlphaFold. So since the 90s, there's been a lot of computational effort to figure out the three-dimensional structure of proteins. This is typically a very computationally intensive task. So you might have seen people in the late 90s, early 2000s with a screensaver called Folding at Home. So in Folding at Home, there was a central computer that would send out tasks, small tasks that idle computers around the world could do, and then send back information in order to piece together a 3D structure like this. So in several months, DeepMind did all of that with one of their newest releases, AlphaFold. So AlphaFold was a real breakthrough in our ability to visualize these proteins, including showing structure like this sort of massive string, this long string around the central structure. In many of our previous perceptions of proteins, it did not include such detail. So here's another view of BRCA1. BRCA1, the gene, it's around 126,000 base pairs long. Here I'm showing the gene. Each pixel here is a different base pair. So the C's are green, the T's are yellow, the G's are blue, the A's are red. So this image is the gene, a view of the base pairs in the gene that someone would usually have. And at the beginning for BRCA1, we have CTT, according to a sequence I obtained from the NCBI, the National Center for Bioinformatics. So this means the first pixel that the color of the first pixel someone would have for BRCA1 is yellow for T, or green for C. But not everyone has the exact same picture here. That's where risk factors come in, genetic risks. So at around 500 locations in the BRCA1 gene, some people will have some colors and other people will have different colors. The two biggest risk factors for BRCA1 discovered in late 90s are 5382 in C and 185 del AG. So these are names of mutations. The names work like this. The number tells you where to look in the gene, offset from the upper left or offset from the beginning. And then ints means add another base pair or insert a pixel here. So 5382 in C says, look here and add an extra green pixel. So that's the meaning of mutation. For the rest of the talk, I'm going to refer to 5382 in C as the first mutation, 185 del AG as the second mutation. But overall, there's around 500 known mutations for BRCA1 that are genetic risk factors. But what does it mean to be a genetic risk factor? In the case of breast cancer or all cancers, genetics is in terms of risk and not absolutes. So just because someone has one of these mutations, one of these 500 mutations, does not mean that they will get breast cancer. So here's an analogy. So this slide was made by, or this image was made by Dr. Rachel Dinchong, who's a genetic counselor at Kingston General Hospital. This jar, when this jar is full, someone can get cancer. And there's two things that can go into the jar: genetic factors and environmental factors. Here we have two people, person A and person B. At the beginning, they're born, but they're born with a certain amount of genetic factors because these mutations are inherited from parents. So in this case, person B's genetic risk is larger. They have more genetic factors already in the jar when they're born. Person A has less risk. They have fewer genetic factors when they're born. But over time, environmental factors are added to the jar. And so cancer can come after the jar is full, after time, and the amount of sort of room for the environmental factors. This is what's changing between person and person. So it's well, and we might ask, what are these environmental factors? So the top environmental factors for breast cancer, according to CDC, are alcohol and not being physically active, and then there's many other environmental factors as well. But if we speak of genetic risk, according to the charity breastcancer.org, the average woman in the United States has about a 1 in 8 or 12% risk of developing breast cancer in her lifetime. Women who have a BRCA1 mutation or BRCA2 mutation or both can have up to a 72% risk of being diagnosed with breast cancer during their lifetime. So the amount of genetic risk circles that are already in the jar can vary a lot, but it's never 100%. And how do we discover that something's a risk? So here we have the first mutation and the second mutation. In this work from mid-late 90s, they proved or they found strong evidence that these mutations were associated with breast cancer. And this is done through statistics. What they do is they look at the frequency of the mutation in people with breast cancer, and they look at the frequency of the mutation in the general population. So for the first mutation, we see that the frequency of the mutation in breast cancer, in people with breast cancer, is 1.9%. And in the general population, 0.13%. So we see a difference. The mutation is a lot more prevalent in people with breast cancer. However, it's still not incredibly prevalent in people with breast cancer, like 1.9%. much less than 100%. So it's certainly not that everyone with breast cancer has this mutation. In fact, only a small proportion of people do. But the difference between these percentages is statistically significant. So in statistics, we can look at two frequencies and say, did that just happen by chance that the frequencies are different? Or are the frequencies far enough apart that it's almost impossible for it to happen by chance? So what does almost impossible mean? This can be sort of operationalized by the idea of a p-value. So a p-value in statistics says, if we look at these two frequencies, the chance of them being so different just for no reason, just because of random chance, the chance of them being so different or even more different, that the probability of that happening is around, in this case, 1 in 1,000. And then in this case, 1 in 100,000. So what this is saying is the frequencies would be this different just by chance, only 1 in 1,000 times. So that's why at first I said that they provide proof, and then I changed that to say they provide strong evidence, because in statistics we're always finding evidence against something. So we never prove something conclusively. Instead, we say the chance that it's not true is very, very small. How small? 1 in 1,000 or 1 in 100,000. So this is what we mean by association. We mean the chance that these frequencies of co-occurrence are the chance that these frequencies could be different just by chance is too small. So I've talked about breast cancer, and I've talked about these two specific mutations, but BRCA1 is only one gene. How do we look at the whole genome? So this is done through genetic sequencing. All human beings are 99.9% identical in their genetic makeup, according to the National Institute of Health. So if you go back to that picture of BRCA1 with all the pixels with different colors, 99.9% of those pixels are going to be the same, more or less, between one person and another. So we can develop we can sort of write down the DNA that's common to everyone, and this is going to be a reference genome, and then the mutations are going to be differences from the reference genome. This is an effort that was first undertaken in around 2000. This was called the Human Genome Project. The Human Genome Project was completed in 2003. So at that point, we heard on the radio that the human DNA had been sequenced. So there's two sort of catches with that. So one is that what the DNA of a single person was sequenced. So for another person, 0.1% of their genome is going to be different from that single person who is sequenced. So the other catch is that the sequence that was produced in 2003 only had 92% coverage. So they only actually sequenced 92% of the genome. The missing regions are called heterochromatic regions. These include telomeres and centromeres. And there's an update to that. So in 2021, the T2T consortium finished with essentially 100% coverage. So T2T consortium, this stands for telomere to telomere consortium. So this is interesting. It could, in some view, the first human genetic sequence and was actually produced in 2021, not 2003. We were missing. We were still missing 8%. These telomeres and centromeres, they occur at the ends as well as in the middle of the chromosome. The telomeres and centromeres do not have many genes, and they actually have a lot of variability between people. So two people will, for example, have different lengths of telomeres. The amount of DNA, the amount of yellow DNA at the end of the chromosome is quite variable from person to person. So for these reasons and for other reasons, it's very difficult to sequence the heterochromatin. And also, especially in 2003, it wasn't really considered a priority. We knew there were no genes there, and we didn't think there was much more than structurally those parts of the chromosomes were doing. But that view has been challenged in recent years. So it is nice that we now have 100% coverage. So this is what we need to do a sort of discovery study where we can look at the whole genome and figure out where in the whole genome, not just in the single gene, there are mutations that matter. So we need sequences like this. So it took until 2003 to sequence one human, but since then the speed and technology has gotten a lot better. And what we're looking for when we sequence multiple humans is differences between their genomes. So these are the mutations. Most mutations are inherited. So the first mutation from BRCA1, the 5382 in C, if someone's parent has that, they may have that. If they receive a gamete that has it. However, in addition, for each fertilization event and a generation coming from that, there's around 20 novel mutations. So this is a really small number compared to the total number of mutations. So essentially, we have inherited mutations, of which there are hundreds of millions of possibilities of possible hundreds of millions of locations where there could be a mutation or not. So the total number of combinations is just staggering. But in addition to these inherited mutations, there can be new or novel mutations that are occurring for the first time in a generation. But the number of these is quite low. And also, it's important to know a mutation isn't necessarily good or bad. Mutations cause variation. So mutations are responsible for the diversity and the for every type of process that humans have biologically. So not just disease, can be anything. So just the word mutation, we might think of that as having a negative connotation, but it certainly doesn't. It doesn't have to be good or bad. In fact, often mutations have no effect physically. So these are called neutral mutations. And there is a theory early in genetics that most mutations are going to be neutral. There's two main types of mutations. So there can be small mutations. If we think of the picture of BRCA1 with all the pixels, a SNP, single nucleotide polymorphism. Take one pixel, change its color from blue to green, for example. So indel, take a pixel and repeat it multiple times, or take a small chunk of pixels and then repeat it. So these are really small, really local in effect. In addition to small mutations, there's large mutations. So copy number variation, a big section of the picture is repeated. Inversion, so the part of the picture is reflected. Translocation, part of the picture moved from one place to another. Large mutations are quite rare. So often in these in studies to make links between genes and phenotypes, we're just looking at these small mutations. And why are we doing this? So part of this is to advance basic science, just to know what genes do, but we can also use this to discover potential therapeutic targets. So if through a study like this, we show that a gene is associated with breast cancer, for example, then maybe doing something with that protein will help with breast cancer. So this genome-wide association study can help us figure out where, can help us figure out what to look at. So what genes should we look at? We should look at genes that have an effect. Which genes have an effect? And then we get that list, and then we can start to work through it and try each one as a drug target. So a final thing for a final reason for doing this is personalized medicine. So especially in the case of breast cancer, there are some drugs for breast cancer that work with some mutations, but not with others. So if we know the profile of someone's mutations, then we can make a prediction of how well they'll respond to different drugs, and then that can be used to make a decision. And this last point here, that this will become useful later. So before, when I was showing the p-values for the evidence of association between the two mutations and the breast cancer status, we were treating breast cancer as a discrete phenotype. Someone either has breast cancer or they don't have breast cancer. So that's just to determine what row of the table to put them in. But these methods can also work with continuous phenotypes. So so a continuous phenotype is something that's on a scale, instead of just being yes or no. Breast cancer, yes or no. A continuous phenotype, we'll see later, it could be something like the strength of the connection between one brain area and another. So this idea of association can be extended to continuous phenotypes. The next step is getting the genetics, because I've discussed that in order to do a GWAS, we must sequence people's DNA. A popular way of doing research in genome-wide association study is by using data from consortiums. An example of a consortium is the UK Biobank. This is a prospective epidemiological study in the UK. In the UK Biobank, 500,000 participants across UK were recruited to be involved in this study. This study involved genetic sequencing. So all 500,000 participants had genetic information recorded. And in addition to that, they had primary care data recorded from their GPs. And a certain subset of them were brain imaged. So in 2020, 40,000 of the participants had brain imaging done with MRI, magnetic resonance imaging. And in addition to that, there's much survey data. So we have information about these participants' diets and environmental status. And going back to the primary care data, this includes information about disease prognosis and treatment. So this study, the UK Biobank, is not about a particular disease. Like this study does not have a hypothesis. This study collects data and then provides that data to researchers. These researchers have hypotheses, and these researchers have specific objectives. The UK Biobank objective is to provide data to researchers. So this UK Biobank, this is how we can do genome-wide association study without having to collect the data ourselves. It is very difficult to get large data of this quality. So to have a single resource, which is then used to answer many scientific questions, is very, very powerful. And also, as I mentioned, this is a prospective study. So what this means is that there's follow-ups. So at regular intervals, since UK Biobank data was first released in 2010, we get updates about the participants. And when COVID-19 pandemic came, UK Biobank could respond by starting to collect outcomes and also conducting serological survey in which antibody status was recorded for participants, and also collecting hospital inpatient data about COVID-19 cases, and also about prioritizing brain re-imaging for COVID-19 positive samples. As the UK Biobank is prospective, we have these follow-up times in which new information about the participants is recorded. Part of that new information is brain imaging. So the UK Biobank has a mandate to re-image re-image participants that have already been imaged. So this can help us understand aging in the brain. So we can see how the brain changes over time, and then use that as the phenotype of a genome-wide association study or GWAS. And in addition, when repeated brain imaging was begun to be conducted by UK Biobank, COVID-19 participants that had COVID-19 were preferentially included in that reimaging. So here, let me talk more about a GWAS on COVID-19. So this is my first example of a GWAS. COVID-19 is caused by the RNA virus SARS-CoV-2. So COVID-19 has its own genetics. I've been speaking in the previous slides about human genetics. So there's 3.1 billion base pairs. That's in the human genome. There's 23 chromosomes, 22 non-sex chromosomes, and then a pair of sex chromosomes. So that's in the human genetics. But in addition to that, COVID-19 has its own genetics. So this is the RNA. The length of the COVID-19 genome is around 30,000 base pairs. And here I've drawn an image similar to the BRCA1 image, where it's 1 pixel per base pair. And again, I got this sequence from the National Center for Bioinformatics, and this is the original strain. So the first SARS-CoV-2 sample to be sequenced. And as an RNA virus, we have U, a small technical thing. We have a U instead of a T here. So this is not what we talk about when we talk about host genetics, and when we talk about doing sort of a human genome-wide association study of COVID-19. So we're not looking at the virus RNA. We're looking at the human DNA. and saying what genes in the human DNA can modulate the severity of COVID-19 or modulate the susceptibility to COVID-19 infection. And there's been much work on this. In a worldwide consortium called the Host Genetics Initiative, a meta-analysis of 81 studies has been put together to find this human genetic signal for COVID-19 severity and susceptibility. So again, as infectious disease caused by a virus, COVID-19 is not caused by human DNA. However, human DNA can contribute to the severity of a case, or it can contribute to the extent to which someone is susceptible. And this is shown through host genetics. So host genetics refers to looking at the genetics of the organism that's infected by a virus, the host, in this case, human. This Host Genetics Initiative is a global consortium to meta-analyze COVID-19 host genetic studies. So one of these 81 studies is UK Biobank. Another one of these 81 studies is 23andMe, the commercial entity in the US that does direct-to-consumer genetics. So with these large organizations contributing to data, last year we had a release from HGI, Host Genetics Initiative, with 290,000 COVID-19 positive cases and millions of population controls. So these population controls have unknown COVID-19 status. And here's the GWAS. This is the genome-wide association study for COVID-19 hospitalization versus population. So population means people with unknown COVID-19 status. Hospitalized means someone who was hospitalized because of COVID-19, according to the hospital that recruited to the study. So the first release from HGI was in summer 2020 with 1,300 samples, and the most recent release last summer with a very large number of samples and many locations on the genome where an association with this hospitalized versus non-hospitalized, or hospitalized versus population contrast is given. So in interpreting this plot, this plot is used a lot in GWAS. This type of plot, it's called a Manhattan plot. The reason for the name is it looks kind of like the Manhattan skyline with some really tall skyscrapers, some kind of medium ones. And on the x-axis, we have the chromosome number. So here's chromosome 1. Here's the beginning of chromosome 1, the end of chromosome 1. And nice, you can see the centromere. So often in a Manhattan plot, you'll see this little dip in the very middle of the protein, or in the very middle of the chromosome. The reason for that dip is because that's where the centromere is. And as I mentioned before, until 2021, we only had 92% of the human genome. In particular, we didn't have the centromeres. And so when DNA was collected from UK Biobank and so on, in sort of the first instance of presenting that back before the T2T consortium was finished and so on, the information from the middle was not there. So you see it again in chromosome 9. The centromere is missing. And also in chromosome 16, you see a little dip here. But in general, Manhattan plot, the x-axis is chromosome. By 23, we mean X, the X chromosome. And on the y-axis, you have the strength of the association. So the higher the value, the better. or the higher the value, the stronger the association. And there's one dot here for every mutation. So here we have hundreds of millions of mutations, and some of them have really strong associations, and some of them don't have a really strong association. And this red line is a level where we say, if the association is higher than that, it's significant. So all of these are different genes or different mutations. Everything above this red line are different genes that are associated with severe COVID-19, and so they are all potential drug targets. So things that we could look at and say, is this gene truly contributing to the severity, or is it a false positive, or is it like a non-causal association? So this concludes my first example, where I was discussing COVID-19 host genetics. So in my second example, I'll discuss brain imaging genetics. So this sort of work is often done with UK Biobank, and it's a line of work that I've contributed a lot to. Here we have MRI images of participants. So back in 2020, we had 40,000 brain images from UK Biobank participants. Each of those brain images is a three-dimensional volumetric image. So there's 1 pixel per millimeter cubed area volume of the brain, and each pixel shows the intensity of the MRI signal, which can give us a view about how many neurons are at a particular low at this like really particular spot in the brain, or it can tell us how much brain activity is there in the case of so-called functional magnetic resonance imaging. So there's many settings, there's many different ways that the scanner gets information about the brain and takes these pictures. So these have names like T1, structurally weighted imaging. So all these different types of imaging tell us about density of neurons, the brain activity, the direction, the direction that water is flowing along axons. This is something that's done by DMRI or DTMRI, or diffusion tensor magnetic resonance imaging. So all of these aspects of the brain imaging give us phenotypes. They give us numbers. So we can sort of quantify from these images the size of a certain brain area or the thickness of a certain region of the cortex. So this gives us one number per patient. So this is the continuous phenotype. So as I mentioned before, just like we had the contingency table for breast cancer, we can extend that to a continuous version. We don't just need to do a GWAS over something that's yes, no, like yes, severe COVID, no, not severe COVID, or yes, breast cancer, no, no breast cancer. Also, when we have an image like this, where the pixel values lie between, say, 0 and 255, as they often are after normalization, then we can proceed and produce the same sort of Manhattan plot. This is work done in collaboration between me and the Wellcome Integrative Neuroscience the Wellcome Center for Integrative Neuroscience at the John Radcliffe Hospital in Oxford University. So the group, a team there led by Professor Stephen Smith and Professor Gwen Duaud have worked with me to do this GWAS on these 40K UK Biobank participants who have been brain imaged. And here's an example of the results we get. So this is the a GWAS on the volume of the left pallidum. This is a brain structure that's right there, right here. It's a subcortical brain structure that's part of the basal ganglia. So it's responsible for some aspects of motor control, and we see that in chromosome 8 there's a mutation that's going to affect the size of this particular brain area. So moving on to the third example for GWAS on brain aging. So this is more work that I've supported from WIN, the collaboration in Oxford. So in this work, they looked at everything in the brain and tried to say, as the brain changes with age, what things change together? So can we sort of take all the brain phenotypes and put them into bins and groups that are potentially aging at different rates? And when this was done, they found 6 big modes. So they found 6 different ways that the brain ages. And so what one is through changes in the cerebral spinal fluid and ventricle volume. So the ventricles, so we have image of ventricles here. So the ventricles are a space devoid of neurons in the middle of the brain and containing cerebral spinal fluid between the 2 hemispheres. So these ventricles can increase in size with age, and at the same time, the amount of cerebral spinal fluid can increase. So that's mode 1. In mode 2, the amount of gray matter, so the amount of the amount of neurons in the cortex can decrease. So these 2 things are not directly related. So it can be that the ventricles increase in size without the gray volume decreasing. So this was a novel result from this 2020 work. And what my contribution to that was looking at the genetic profiles for these different modes. Here we have one last Manhattan plot here. Each of the 6 modes is a different color. So we did sort of a genome-wide association study on how much aging there was in each of the modes. And we find, for example, mode 5 is associated with a chromosome 11 mutation, and mode 4 is associated with the chromosome 5 mutation. So broadly, this work is showing that the aging brain is not driven by a single process. It's driven by at least 6 processes, and these processes have a different genetic makeups. So I'm going to finish with this third example. So much of the work I've shown you is work that I've contributed to. For example, the Host Genetics Initiative. I'm a member of the Host-Seek collaboration. So this is Canada's contribution to the Host Genetics Initiative. the HGI, as I mentioned, is made up of 81 studies. One of those studies is HostSeek, which I worked with to collect data from across Canada on. So I talked about work that I supported on COVID-19. So that's done in collaboration with Magpie at SFU. And I have more COVID-19 epidemiological work with University of Victoria, or UVic. So here are my students and colleagues in the stats department that have contributed to the work that I've discussed. And Olga Vishnyakova is also on this list since I last updated this slide. And the WIN collaboration is with Oxford. And my main funding is from NSERC and the Michael Smith Health Research BC. And for COVID work over the past 3 years, I've also gotten grants from Genome British Columbia and Genome Canada. So thank you all so much. And with that, I'll move to questions.
Thank you. Thank you so much, Lloyd. At this time, I would like to invite the audience to keep typing their questions or comments by using the Q&A box. We appreciate your sharing, Lloyd, this presentation, and I personally like the last slide where you described all your collaborative work. That was awesome. Lots of things going on. Very interesting research that you had presented. There are a few questions that are coming up on the Q&A box, and I'll start with the first one. And this might be pertaining to that last slide, actually, where you described other projects and collaborations.
Mm-hmm.
What is SFU's role in this research?
Oh, right. So I started at SFU in 2018, and after arriving here, I was really lucky to continue the collaboration with Oxford. So from SFU, I've added some of my trainees to, over the years to work on brain imaging, in particular Sunny Min, and also Winfield Chen, alumni from my lab, have worked in collaboration with Oxford. And also on the epidemiological side, SFU has really strong participation in modeling for COVID-19. So this is with Caroline Colleen, with other professors in stats like Liang Liang, and also many projects here in stats that I'm not involved in. So SFU's involvement in this is definitely strong in many ways, and I would say one of the biggest ways is the impact that we've had for modeling and preparedness. So this is part of the reason that many at SFU were invited to be involved with PIPS, the Pacific Institute for Pandemic Preparedness. So and also on the HostSeek side, there is some sample collection in Vancouver for HostSeek, but SFU is not directly involved in that. However, some of my trainees at SFU have contributed to this collaboration.
Well, thank you. Thank you for clarifying that. Someone in the audience is asking, this is very interesting, why are the genes BRCA1 and 2 called BRCA? Yep.
Yeah. So here it's sort of an acronym. So this BRCA is an acronym for breast cancer type 1. BRCA1 is an acronym for breast cancer type 1 susceptibility protein. it's kind of interesting because it's a gene, not a protein. However, the gene codes this protein. So not all genes code proteins. So sometimes you'll see in sort of the expansion of a gene, things don't always line up, but essentially the BRC in BRCA is breast cancer.
Mhm.
And the job of the protein is to repair DNA, destroys cells with DNA errors. So it's not necessarily to fight breast cancer, but for breast cancer, this BRCA gene is particularly important. This BRCA, scientists came up with that name as they discovered the link between breast cancer and this gene. But you will often struggle to see sort of the one-to-one correspondence between the letters in the acronym for a gene and the full name of the gene or the name of the protein that it codes. Part of the reason for that is just the large number of genes in the human genome.
Mhm. Thank you. Now let's head over to that slide with the a circular brain diagram. Someone wants to know, what does this diagram mean?
Yeah, so these are these are connection strengths between brain areas. And this was found during fMRI, or so-called functional magnetic resonance imaging of the brain. So for this fMRI data, the UK Biobank has someone remain in the scanner for around 15 minutes. So they look at their brain activity over 15 minutes. And they sample it at some frequency. So it's like they have one volumetric image of the brain activity per sampling occasion. So they look at this over time, and they ask how often does activity co-occur in two brain areas.
Mm-hmm.
And this co-occurrence is indicated by these lines here. So a single subject will have different thicknesses of lines.
Right.
These brain areas, these are sometimes called networks because they're not necessarily a contiguous region of the brain. Like here, the 22 is like, this looks like this looks like temporal lobe, both left and right. So it's not a single brain area. It's the same brain area on the left and right. But to have this line between like 22 and something else means that these two brain areas often are activated at the same time for a participant of the UK Biobank.
Wow. Someone's interested in going back to that third slide from the end on brain aging. If we can look at that again. And her question is regarding modes and aging brain. Could you identify the main indicators?
Yeah. So main indicators, I guess I've cut off this table. There's 6 more mode clusters. And for each of the mode clusters, we've looked at the participants of the UK Biobank and looked for phenotypes in the brain that tend to change together. For example, we found that with age. So we found that as someone ages, the amount of change of cerebral spinal fluid is sort of proportional to the amount of change of the ventricle volume. But the amount of change of these two things is not necessarily proportional to the change of gray volume. So each of these modes has many brain phenotypes that are changing together. Within a cluster, they change together. Between clusters, they change independently. So this means as someone ages, maybe their CFS ventricle volume does not change much. However, their gray volume could change and decrease. Or it could be the other way around. The gray volume stays more or less the same, but the CSF ventricle volume increases. If CSF increases, ventricle volume will tend to increase because these phenotypes are in the same mode.
While we're speaking about brain age, I'll read the next question that I see connected to that. Someone's wondering if you can briefly go over how estimated brain age and actual age tie into the brain imaging and research, such as that formula used in the Smith 2020 paper.
Yeah, so the main idea there is we so we use the phenotypes to estimate age. So we're essentially trying to in the first step of analysis, we have the age of the eye patient or the eye participant, and then we're going to we're going to express that as a weighted sum of a weighted sum of the brain phenotypes. So here's the age, and we're trying to find weights that work for all participants. So every participant will have the same way of weighting their phenotypes. So what this means is with this prediction of age from brain phenotypes, we're not going to exactly recover their age. There's going to be a bit of an error. So if we have the predicted age like this, then their actual age is AI. So this is the delta. So the brain age delta, this is the difference between someone's actual age and the age that we would predict based on their phenotypes. So this brain age delta is the target of the genome-wide association study. So essentially that's how it's used.
Awesome. The person who asked that question appreciated that answer, so thank you so much for that. I'll read the next question. How can statistics and genetics be used to understand connectivity between brain regions?
Okay, so this question is similar to the network question. We're already using statistics in order to produce these networks. So if we're asking how much is network 22 connected to network 41, for this, we would look at a statistical correlation within the fMRI data. So statistics is used in a very simple way to construct these weights, just by looking at the essentially Pearson correlation between the two networks in the sequence of fMRI images. But moving to genetics, the way that we use genetics to understand this is through the genome-wide association study. So we want to make a one of those Manhattan plots. So we have chromosomes 1 until 22 in X. So for each gene, we want to find the strength of the association for something, for a continuous phenotype. There's, for each subject, by looking at this correlation over the fMRI signal, for each pair of networks here, for like 22, 41, there's a strength. So it might be something like 0.1. So one participant, make a red line here. So 22 is connected to 41 for a particular participant. And so that say participant 4 has a 22-41 connection of 0.1. So this is the continuous phenotype.
Mm-hmm.
So participant 5 will have a potentially different number for 22-41.
Mm-hmm.
Participant 6, a different number for 22-41. So this will serve as the continuous phenotype in the contingency table that we make for each gene, which will then give us an association number. So in the end, we'll get a Manhattan plot. So if it looks like this, the title is 22 versus 41. So for this particular link, we find a single Manhattan plot. So we use genetics to know that, to understand this link by seeing if there's any hits, because if there is a hit, like if there is association that's above the solid red line, then that means genetics plays a role in the link between these two brain areas.
Right. Interesting. Wow. Speaking of genetics, if mutations are inherited, you mentioned that mutations can be inherited. Is it possible to predict this before a person is born? And is it possible to repair this if it happens to be a bad mutation?
So this is a topic in genetic counseling. So in particular for so-called Mendelian diseases, that this is definitely an important aspect. Genetics for most cancers is in terms of risk and not absolute, but this is not true for every disease. So for so-called simple Mendelian diseases, then for example, if if we inherit 2 copies of the effect mutation, then the condition can always come. So I believe ALS is an example of that. So in this case, we might want to go to a genetic counselor to know the probability that an offspring would have have the condition, and with the guidance of doctors and genetic counselors, then the situation can be navigated. But in terms of other in terms of other situations, so think about a situation that isn't Mendelian. In that case, it's certainly possible to predict things like well, it's on this slide, risk of cancer for the next generation. This is something that could be approximately found by looking at genetics, but at least how genetics is now, we don't have, in my opinion, we don't have enough accuracy to sort of personally act on that, even if we did want to. But in terms of the in terms of ethical considerations about things like that, my work is about the science, about basic science, discovering therapeutics and personalized medicine. So I don't have research that speaks to the specific details of your question.
Right. But I appreciate that slide that you just showed us, the one with the bottles. And I just wanted to read out that one comment that one of the people in the audience had said: very awesome presentation. Bravo. Thank you for sharing with such simple explanations. And visuals like this really help us understand, and illustrations like this are very helpful.
This illustrate, well, speaking of genetic counselors that can help us navigate such difficult decisions, this slide was made by Dr. Rochelle Dinchong, who's a genetic counselor at the Kingston General Hospital. So this particular jar analogy.
Right. Someone in the audience was also wondering if you are going to be sharing your notes or your slides. I will say that we don't usually share slides and notes, but we record the event, and you're happy to review the recording. It will be posted on our faculty YouTube channel. So you're welcome to share it as well, because it will be made public and shared broadly on that YouTube channel. I'll end with this last question, Lloyd, if you don't mind. Just looking through the whole Q&A box if I missed anyone else's. Okay, I'll just read this last one. It seems like you would need to pick up a lot of domain knowledge when you work on a new area. Is that right? And is that difficult? And is this a barrier to how many projects you can take on? You've mentioned several projects you've contributed to.
Yeah, it's a good question. So for a trainee, I think the requirements of domain knowledge in specific applications of genetics is not a difficulty, especially for trainees in statistics. A reason for that is, so I've discussed a very simple statistical test here, but some of the tests and procedures that are used statistically in genetics are quite complicated. And in the end, they may boil down to an equation that must be solved. So a trainee can appreciate that equation, and then focus their energy just on that. So like develop an inference method for this particular equation. Here's the equation, here's the free variable A, find A. So this is something where a trainee can make a big contribution without having the domain knowledge. And then if they're part of a larger collaboration, like if this was a task that was assigned to them in order to make a way of analyzing new data, then they can sort of come together with that collaboration, have their contribution to it without at that time requiring the domain knowledge. But as the trainee progresses, if they continue with the field, then the domain knowledge will come. And that's something I've seen in my own experience. So I am a mathematician by training in undergrad, and then I did machine learning in my PhD. But early in my PhD, I started with genetics, genetics applications, and so that's something that I built over the years, and I never felt it was a barrier for myself learning the kind of genetics that I've shared with you today.
Right. And I'm glad that question came in. There may be some high school students in the audience who are lovers of math and statistics, and this is a perfect way of illustrating how math and statistics can be used in genetics and biology and so many other things. So thank you so much for sharing your research with us. And you've shared it in such a simple and visual and easy to understand manner. And we appreciate you're doing this for us tonight. Well, thank you so much, Lloyd.
Thank you. And I'm so glad for all the questions.
Yes. And we're happy that everyone has contributed their questions and comments. Thank you so much. We apologize for those who have typed in something and we have not had time to read everything, but we certainly appreciate your engaging with us. Thank you for joining us tonight. And thank you again, Lloyd, for being with us. This was our fourth and last spring Café Scientifique session. We will be back in the fall term. I hope you can join us then. You're all part of our mailing list and you will be receiving updates on the next fall events. I hope you can join us then. In the meantime, enjoy your spring term and your summer months. Lots of sunny weather ahead, I hope. In the meantime, stay safe and enjoy the rest of your evening. Thank you so much, Lloyd.
Yeah, thank you.
You're welcome. Bye for now.